Breakthrough, improved CRISPR gene drive solves problem of old tech. I'm Steve Belair, welcome to Breakthroughs. Gene drives use genetic engineering to create a desired mutation in a few individuals that then spread via mating throughout a population in fewer than 10 generations. In theory, such a mechanism could be used to prevent malarial mosquitoes from transmitting disease, or possibly to wipe out an invasive species by disabling its ability to reproduce. Though scientists have had success proving the concept in the lab, they have found that wild populations invariably adapt and develop resistance to the scheme. And when gene drives work, they are all or nothing. Without nuance, they spread to all individuals, which can be a drawback. Now a Cornell study, a toxin antidote CRISPR gene drive system for regional population modification, published February 27th in the journal Nature Communications, describes a new type of gene drive with the potential to delay resistance. The method could also be applied to regional population, limiting its spread to other populations where it could have undesired effects. Those are two things that this new drive that we've developed have addressed to some extent, said Philip Mazier. In a classic gene drive called a homing drive, an offspring inherits one set of genes or genome from the mother and another from the father. If an offspring inherits a gene with the drive from one parent and not the other, the drive copies itself into the genome from the parent without the drive. Now that individual has the drive in both of its genomes and it will pass it off to every offspring, Mazier says. The drives are engineered with CRISPR-Cas9 gene editing technology, so when the drive copies itself into a new genome, the CRISPR machinery makes a cut into the chromosome without the drive and pastes in the new code. But sometimes cells will repair the incision and in doing so randomly delete DNA letters. When this happens, the CRISPR gene drive can no longer find a genetic sequence it recognizes in order to make the incision, which creates a resistance and stops the gene drive from spreading. Neurogenetic variation, another source of changes in DNA sequences, can also create resistance, since CRISPR gene drives must recognize short genetic sequences in order to make incisions. We were among the first labs to show that this is a tremendous problem, Mazier says. The paper describes a new gene drive called a TAR, toxin antidote recessive embryo, which works by targeting a gene that is essential for an organism to function. At the same time, the organism can survive with only one intact copy of this essential gene. Instead of cutting and pasting DNA as homing drives do, the TAR drive simply cuts the other parent's gene, disabling it. Meanwhile, the engineered TAR drive gene has a DNA sequence that has been recoded. The gene works, but it won't be recognized or cut in future generations. If an individual inherits two disabled genes, those individuals won't survive, thereby removing those copies from the population. Meanwhile, as viable individuals mate, more and more surviving offspring will carry TAR drive genes. Just a few individuals with homing drives can spread a trait through an entire population. TAR drives, on the other hand, do not cut and paste the drive into the target gene. Instead, they destroy one of the target gene copies in the offspring. Because of this, the drive requires a higher frequency of engineered individuals in the population to spread. For this reason, TAR drives are less likely to transfer from one distinct population to another. Wow! I'll leave a link in the description to the full article, and as usual, please like, share, subscribe, and take care.